Welcome back to Bio 111. This is Chapter 13, The Modern Understanding of Inheritance. If you recall from last chapter, we talked about Gregor Mendel and his work with pea plants. He used these pea plants, or he crossed them and observed the patterns of inheritance that he saw in the color of the flowers, the color of the peas themselves, the texture of the pea seed coats. And from those observations, he was able to create some laws. He published his work in an obscure journal in 1866, and his work was largely ignored until 1900. And that's when four scientists independently rediscovered his work after duplicating his findings. Now, one thing to note is that Mendel did not understand the physical mechanisms behind his laws. He simply observed the results of his crosses and created rules to explain them, or rules to predict them. If you remember, we talked about that he had this conception of a hereditary particle. He said it was being passed from parent to child. And these scientists, to follow him, that rediscovered his work, they took on the task of trying to determine what those particles really were and what the mechanics underlying these laws were. Right? Now, by 1900, we had a new tool under our belt, the microscope. And you'll recall from our meiosis and our mitosis discussions that there is a particle that is visible in a microscope that is responsible or part of the hereditary process, and that structure is a chromosome. Now, you'll also recall that chromosomes are made of DNA, and DNA is the data storing molecule inside of cells. That is where we have all our information that determines our phenotype. Now, these scientists, when they discovered chromosomes and began to link them to or claim that they were the hereditary particles, they were really not doing anything different than Mendel. They were just looking at it microscopically. They were essentially saying these particles move from one generation to the next generation during crosses, following the same laws that Mendel discovered. So they essentially just found that there were particles inside the cell that obeyed Mendel's law. All right. Walter Sutton and Theodore Boveri are credited with developing the chromosomal theory of inheritance, which states that chromosomes carry the unit of heredity, genes. They found that during meiosis, chromosome pairs migrate as discrete structures. Each parent synthesizes gametes containing only half the chromosomal complement. Even though male and female gametes, sperm and egg, differ in size and morphology, they have the same number of chromosomes, suggesting equal contributions from each parent. Right? Genetic or the gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. So remember, from meiosis, we end up with haploid cells. The sperm and egg are haploid, meaning they have half the number of chromosomes as the parent. And then once they recombine during fertilization, we have a diploid cell again. All right, that's what's just being said there. Now, again, I said that they really did just the same thing that Mendel did as far as looking at patterns and understand and recognizing that these chromosomes followed Mendel's patterns. They didn't really have any direct linkage between or any mechanism for how these chromosomes were actually doing anything to carry out these, to how it mapped, how these chromosomes map to the phenotype. All right. Now, that changed the discovery of X and Y chromosomes, because you could show with this that there was a direct link between male-female and the presence of the X and Y chromosomes. All right, that's the first real proof of chromosomes' role in heredity. T.H. Morgan showed that white eye mutant allele was inherited along with sex, right? And this sex linkage was the first demonstration that genes were on chromosomes, right? Genes, this being, the hereditary particles. The concept of linkage was further extended to map genes to chromosome positions, genetic linkage maps. So the idea is that you could determine where on the chromosome the genes were relative to other genes. So Morgan's work with sex linkage showed that the trait for sex and the trait for eye color were inherited together. So these are linked genes. They did not segregate independently. This violates Mendel's second law. When other traits were examined, it was found that those on the same chromosome did not always segregate independently. Traits on the same chromosome did segregate, however. Far apart traits segregated independently. Traits closer together segregated less often than the independence predicted. Crossover between homologous chromosomes was a mechanism that segregated genes on the same chromosome. When genes were far apart, crossover happened all the time. When they were close together, crossover happened less frequently.